Hi there, science fans. Welcome to another episode of Science and Other Stuff for Senior Citizens. My name is Dan Casciano. If you like these COVID-19 videos, please subscribe to my channel and ring the bell to be informed of future videos. In this COVID series, I focus mainly on the COVID-19 virus infection pathway and generally how the human responds, and to a certain extent, I discuss health issues that contribute to the severity of the response. In the present video, I will describe 3 very recent research studies that conclude there may be good news on the horizon. I decided to present the conclusions from these studies in this slide. In the following slides, I will present a summary of the data that allow the authors to conclude that long-lived immunity to COVID-19 can be achieved in humans regardless of the severity of the viral infection. The 3 studies show that antibodies produced in humans upon infection by SARS-CoV-2 offers protection to the individual. Prior to these studies, it was known that virally infected animals produced antibodies that protected the animal, but human data were not yet available. Additionally, cellular immunity after infection is long-lasting because of the induction of B and T memory lymphocytes, suggesting that vaccine-induced cellular immunity will be long-lasting as well. This long-lasting immunity was not dependent upon severity of the response to COVID. Even mild cases in asymptomatic individuals resulted in long-lived immunity. All of these data indicate that herd immunity could be achieved. So what exactly is herd immunity? Herd immunity occurs when a large part of a population is immune to a specific disease such as COVID-19. It usually occurs upon vaccinating a large percent of the population. There is general agreement that herd immunity is achieved when 70% of the population has antibodies to that specific pathogen. However, some scientists agree that 50% immunity would slow the spread of the virus, and 60% would slow it even more. 70% is considered necessary to eradicate the virus. This slide is here to remind me to review the 2 virus outbreaks associated with coronavirus severe acute respiratory syndrome. SARS-CoV-1 refers to the coronavirus outbreak that occurred in 2003, while SARS-CoV-2 refers to the present coronavirus outbreak that we are presently exposed to and is known as COVID-19 because of the year of its discovery. This slide is here to remind us of the immune cells that respond to viral infections. Lymphocytes are one of the cell types found in the circulating blood. The lymphocyte population contains 2 types of cells, the so-called big lymphocyte and the small lymphocyte. The big lymphocyte changes or differentiates into natural killer cells. These cells are part of the innate immune system that recognizes virally infected cells and kills them. These cells are part of our first line of defense against viral invaders. The small lymphocyte differentiates it into the B and T cells. B cells mature in the bone marrow while the T cells mature in the thymus. Upon infection by a specific virus like COVID-19, the B cells differentiate into plasma cells that then make the antibodies necessary to neutralize the COVID-19 infection pathway. These antibodies are specific to the invading virus. When our body is exposed to the flu virus, the B cells transform into plasma cells that produce antibodies specific to neutralize that virus type. The T cells, on the other hand, differentiate into helper, suppressor, and cytotoxic cells. The helper cells synthesize messengers that stimulate the B cell to make specific plasma cells and specific antibodies, while the suppressor cells make messages to tell the helper cells to stop producing specific antibodies when sufficient quantities of that antibodies are available to stop the infection process. The T cytotoxic cells identify body cells that are already infected with the virus and proceeds to kill them to inhibit further production of the virus. In addition to these cell types, the T cells differentiate into T memory cells that remember the antigenic signature of a particular virus, like COVID-19, and expands their numbers to stimulate the B cells to produce antibodies to fight the virus. Because of the T memory cells, the body can respond to the invading virus much more quickly. The study presented here was done by scientists at the University of Washington in the state of Washington. The investigation studied whether the presence of antibodies correlated with protection of COVID-19 in humans during a fishery vessel outbreak with a high attack rate. The attack rate is determined by the number of exposed individuals who present with symptoms and/or the presence of the virus. The fishermen were tested for the presence of COVID-19 neutralizing antibodies and the virus prior to departure. None tested positive for the virus, while 3 tested positive for the antibody because of a previous bout with COVID-19. 18 days later, the ship returned to port because 85% of the fishermen showed symptoms of COVID-19 infection. One of them required hospitalization. Upon return, the fishermen were retested, with 104 of them testing positive for the antibody, while 101 were positive for the virus. 3 crewmen showed no symptoms, nor did they have the virus. These 3 crewmen who had recovered from a previous COVID infection were protected from reinfection. These data were the first to show that humans become immune after infection by this virus, suggesting that vaccines may be active in providing long-lasting immunity. This is good news. This study is also a good news story and supports the findings of the study on the previous slide. The investigators studied T-cell immunity in convalescent individuals with asymptomatic or mild COVID-19 and found that virus-specific T memory cells persist for many years after infection with SARS-CoV-1. And these T cells showed high killing activity that correlated with disease severity. Also, SARS-CoV-2 specific T cells were found in antibody-negative family members and individuals with a history of asymptomatic or mild COVID-19 disease. These data indicate that SARS-CoV-2 induces robust T cell memory responses and suggest that these individuals are likely to be immune from future infection with SARS-CoV-2 and should not get the disease. This also suggests that a vaccine would induce these same types of T memory cells. A third study that comes to a similar conclusion as the first 2 studies is shown in this slide. As we know, the majority of infected people experience mildly symptomatic disease, but it is not known whether they experience persistent immune response. In this study, testing was done on individuals who had recovered from mildly symptomatic COVID-19. It was found that these individuals developed SARS-CoV-2 antibodies as well as virus-specific B and T memory cells, indicating that even in mild cases, persistent immunity is developed. The good news is that all 3 studies show that regardless of the severity of the disease, long-lived immunity is achieved. This suggests that herd immunity may be possible and means that vaccines will induce specific viral antibodies and long-lived B and T memory cells and be effective in warding off reinfection. Let us continue to wash our hands, physically distance at least 6 to 10 feet, and please wear a mask. For more information about science, visit my website at www.dcasciano.com. You will also find an animated video of T-cell immunity. Check it out. If you have any questions or would like me to develop a video on a specific science subject, leave them in the comments below or email me at dan@dcasciano.com. Be sure to like this video and subscribe to my channel and ring the bell so you can be informed when I upload a new video. Thank you for watching Science and Other Stuff for Senior Citizens. Bye.